All right, guys, welcome to 6.3. We're going to look at genotype and phenotype. Let's jump into it. Okay, so in the last chapter, we talked about alleles, right? So you remember you have two variations of the same gene, one from mom and one from dad. Now, turns out not all of the alleles that you have are generally expressed in an individual, but the alleles, the two ones that you get from mom and dad, have a dominance relationship, right? So alleles are characterized as either dominant, which means they are always going to be expressed in an individual, or they're going to be recessive, which means they're going to be expressed when the dominant allele is absent. Because of the two copies, each gene has these particular dominance pattern in which one is going to be expressed. And so those dominance patterns can take form in one of three ways, right? One, you have what we call complete dominance. Complete dominance is when the dominant allele is going to completely mask the recessive allele, and the recessive allele is not going to be expressed at all, right? You can also have incomplete dominance, where the dominant one doesn't fully mask it, and you get what we call an intermediate expression of both of the alleles together. Now, this is also called partial dominance. And then finally, you can also have co-dominance, where both of the dominant alleles are going to be fully expressed within the individual, okay? So every gene is going to be different. So the gene for eye color, or the genes for eye color, is going to have a different dominance relationship, which, end up could be co-dominant, for example, compared to that of whether or not your earlobes are going to be attached or, or not, or whether your freckles or the color of your snot or whatever, right? They are all going to have different types of dominance patterns, right? And it's up to each gene to, or up to you to recognize what type of dominance pattern is displayed in each of these genes, right? Okay, now, before we move on, I got to talk to you guys about gene and allele notations, right? When we want to represent an allele, we are going to use a letter, right? The letter is going to represent the gene. The same letter represents the gene, and then the capital of that letter is going to represent the dominant allele, and then the lowercase of that same letter is going to represent the recessive allele. So, for example, right, this is Gregor Mendel. He is basically the father of modern genetics, because he was an Austrian monk who kind of came up with this particular pattern, and even today we call it Mendelian genetics because it's named after him. He studied peas a lot, right? And he noticed that certain traits of peas have, different kind of dominance or preference patterns. So, for example, the dominant trait in most flower color was purple, right? And so purple, with a capital P, is going to be the purple flower color allele, right? Now, the recessive trait for flower color was white. So a lowercase p, notice how it's lowercase p, is going to represent white. So they don't use the same letter for that same gene, but the capital is for the dominant and the lowercase is for the recessive, right? On top of that, the flower position could be axial, means it's along the axis, or it's going to be terminal, which is along the terminal, which is at the end, right? Axial flower position is dominant, so it's going to have a capital A. And the terminal flower position is not going to be represented by T, it's going to be represented by a lowercase a. Now, these letters are arbitrary. I could use X, I could use Z, I could use Y. It doesn't really matter which letter you use. It's just the most convenient one is you usually express whatever the first letter of that word that you're referring to is. So you can see there, C color might be Y, for example, or it could be green. I don't know. It's up to you, right? Now, if it is on the X chromosome, in other words, it is X-linked, then what you do is you actually have to use the letter X, and you use a superscript, a smaller letter in the top position, to represent whether it is dominant or whether it is recessive. So, for example, capital X with a capital A is going to be, that particular allele A for that particular gene. I don't know, whatever the gene is, the DMD gene, for example, right? And then the lowercase is going to be the recessive version of that X chromosome, okay? Now, for males, you can have capital A or lowercase a for the X chromosome, and because a Y chromosome is just on its own, there is no dominance pattern. You can't have capital Y, lowercase y, or YA, or YA, capital YA, lowercase, right? Because there is only one Y chromosome. However, there are different types of X chromosomes, right? Which is why you can still get these combinations. Now, when we talk about genotype, we also, when we talk about genes, we also have to talk about genotype and phenotype, okay? And the term can be quite confusing, but in general, when we are talking about the genotype, we are talking about the set of alleles present in the DNA of an organism, right? That is going to form the genetic makeup of the individual. And then each gene has 2 alleles, which make up the genotype, right? So because of the gene having a particular trait, you know, capital A is going to be a form of genotype, right? Because that is what you would have in your body. Now, you can have different versions. You get capital A, and that is what we call a homozygous dominant genotype. Dominant because they're both dominant letters there, and it's homozygous because it's the same letters, right? Capital A. If you had a lowercase a, that would still be homozygous because they're both lowercase a's, but it will be homozygous recessive genotype, right? Lowercase a. So yeah, so they're slightly different. They're both homozygous, but one's homozygous dominant, one's homozygous recessive. If you have a different combination, however, that in between there, capital A, lowercase a, then you have what we call a heterozygous genotype, right? Where one allele is dominant, but the other one is recessive. And there is no such thing as heterozygous dominant or heterozygous recessive because you have one of each, right? Now, that's the genotype. So if I said, what is the genotype for eye color? You might say eye color would be capital E, lowercase e for, you know, individual A, or capital E for individual E, individual B or whatever, right? And those 2 letters represents the genotype. The phenotype is the actual trait we are talking about, right? The genotype are the genes. The phenotype is the actual eye color, hair color, blood type, genetic conditions. That is the trait that is now being expressed by the genotype, right? In general, we have a formula to help us understand how genotype and phenotype are related. The phenotype, the trait, is the genotype together with the influence of the environment. And we're going to talk later on in this particular video how the environment plays a role in that, okay? So that's that. Now, let's take a look at dominance patterns, right? And you've got your three different type patterns. We're going to start with complete dominance. What happens when the dominant allele is going to completely mask the recessive allele? And we're going to start by talking about cystic fibrosis, right? Cystic fibrosis is a genetic disorder. You are born with it. That's why it's a genetic disorder. And, you know, it is something that is passed on from generation to generation, right? It's going to affect your lungs, pancreas, and liver, kidneys, and intestines. 1 in every 3,000 newborns of North European descent will have cystic fibrosis. And in general, there is a clogging of the airways because there is a mucus buildup and inflammation in those areas. You get a thickened secretion, and that thickened secretion around those, membrane barriers are going to cause damage to organs like pancreas, giving you pancreatitis and diabetes as a result of that. In addition, 97% of men with cystic fibrosis are usually infertile because there is a congenital absence of the vas deferens. The vas deferens is the tube that delivers the sperm from the testes out of the penis. And so men with cystic fibrosis are generally missing that tube, and therefore they are infertile, right? Now, this is your CFTR channel, right? The CFTR channel plays a really important role, right? It stands for the cystic fibrosis transmembrane conductance regulator. Now, bit of a mouthful, right? But it is referring to cystic fibrosis, the condition. Transmembrane, it runs across the entire length of the plasma membrane, and it is a conductance regulator protein. This is a protein, which means the protein has a particular role in the plasma membrane. It's going to regulate the movement of chloride ions in and out of the cell, right? Now, that is really important because chloride ions are going to remove mucus. If you have a recessive allele for this gene, the recessive allele is kind of like a really badly written recipe, and you end up producing a CFTR protein that doesn't work. Because it doesn't work, it doesn't move chloride ions out of the cell to remove mucus along organ membranes. And so things like the pancreas or the lungs are going to have a mucus buildup, and that's going to create issues, right? So this is a mutant CFTR channel. At some point in time, there was a mutation which made it ineffective or non-functional. It doesn't do what it's supposed to do, and then that allele is being passed on from parent to child over time. Okay, so how do we put it together? So it's the CFTR gene is transcribed to form the mRNA sequence and then translated to produce the amino acid that's going to be folded to form the protein, right? So that That's how you get from gene to protein, right? Transcription, translation produces the protein, and if it works, it moves chlorine ions out of the extracellular space and then removes the mucus as a result of that. If you have a faulty one, for example, like this one here, there's a DNA change here in the recessive allele in exon number 4. Don't worry about that. That's not really important, but you get some sort of wrong substitution. And then what happens is the channel doesn't open properly as a result. Now, there's another way that you can get cystic fibrosis, which is another DNA change along exon 10, where there's a deletion. And so the protein's missing a chunk out of it, right? And as a result, it doesn't fold properly either. Either way, you get cystic fibrosis, and if you get this version here, you get the absence of the vas deferens as a result as well, right? So that that faulty genetic sequence, that is found in the recessive allele. When you have complete dominance, the dominant allele is going to mask the recessive allele. So for cystic fibrosis, right, a capital C is going to represent a normal CFTR gene. A normal CFTR gene means that you're going to get functional CFTR proteins along your plasma membrane. That's good. It means it works. If you have a mutated one, you have a recessive lowercase c CFTR gene resulting in a non-functional protein. Now, that means that in any individual, you can get one of 3 outcomes. You either have capital C, lowercase c, or lowercase c. Remember, this one's from mom, this one's from dad, right? If you inherited 2 capital C's from both your parents, you have no cystic fibrosis. You're fine. If you inherit a capital C and a lowercase c, you still have no cystic fibrosis. Why? Because even though you carry that recessive allele, you have a dominant allele that's going to mask that, which means you're still going to produce normal functional proteins, and you're fine. However, if you have 2 lowercase c, you have both recessive, a homozygous recessive genotype, you are going to have cystic fibrosis because you cannot produce normal functioning cystic fibrosis transmembrane reductants, regulated proteins as a result of that, right? So you only need one dominant copy of the CFTR to be expressed, right? And that's good. So in general, those are the possible outcomes. Now, the giveaway is this one here, the heterozygous genotype. If the heterozygous genotype ends up with the same phenotype, the same trait, no cystic fibrosis is the trait, as your homozygous dominant allele combination, your genotype, then that tells you that it is a complete dominance feature. Now, let's talk about another dominance pattern, right, which is present in snapdragons. These are snapdragons. They're very beautiful flowers, and they come in all sorts of different colors. The seeds are funky as because they look like little skulls, and snapdragons exhibit what we call an incomplete dominance, right? Incomplete dominance is when the dominant allele doesn't fully mask the recessive allele. So we're going to use flower color as our gene, and the alleles for flower colors are, you've got 2 combinations, right? You have capital R for a red flower color, and then lowercase r for a white flower color. Now, that means that there are 3 possible outcomes once again, right? You can have capital R, which produces red flowers, lowercase r, which produces white flowers, or you can have capital R, lowercase r, which in this case produces pink flowers, right? So the pigment, the red pigment here, right, is dialed up to max and you get red. The red pigment here is dialed down to 0, so you get no pigment, which is white. And then this one is kind of dialed halfway, and you get pink, right? And so that is what we call an incomplete dominance, right? That capital R doesn't mask the lowercase r, and a bit of the white comes through, and it's not complete, right? It doesn't completely dominant. And so you get a blend of the homozygous dominant and the homozygous recessive allele, right? So that tells you that it is incomplete or partial dominance. All right, the last dominance pattern is codominance. Sorry, that's completely blocked out. So I don't know why that's completely blocked out, right? Codominance happens when you have 2 dominant alleles that are going to compete with one another and both be expressed. In this particular example, we are looking at the coat color in cows, right? So if you have a codominant pattern, we actually use a slightly different notation, right? We actually use the same capital letter, and then we actually end up using superscript notations, just like the X chromosome ones, to represent one. Why? Because they're both codominant and they're both dominant. So you have to use a capital letter for both, but then you're going to use a little superscript letter to represent each of the colors, right? So RR is a red coat, RW is a white coat, right? So you have three possible genotypes in this particular case, right? RR and RR, which is red, RW, which is white, and then if you have RR and RW, you have a Rowan coat, right? A Rowan coat is that typical kind of, dairy cow coat color, right? That patches of red and white, that tells you that that is a co-dominant feature, right? Because you have both being fully expressed. Patches of red here, patches of white here, and as a result of that, both of them are sharing the trait that's being expressed. Now, how is codominant different from incomplete dominance? In codominance, you have a blend. In incomplete dominance, you have a blend of both colors, right? So red and white would have formed pink. So if these cows came out pink, right, that would be in incomplete dominance. Why? Because you're getting a blend of, a spectrum, right, from red to white, and halfway between the colors is pink. However, in codominance, you have both of them fully expressed, right? Which is giving you an idea that, they are both shared and fully expressed as different pigments, right? Now, here's another example of codominance, right? We're going to talk about ABO blood type. Really, really cool thing to understand, and particularly if you ever go into things like nursing or paramedics, and we talk about blood donations and things like that, it's really important that you get the blood type right? Blood type represents what we call an antigen marker that are present on all blood cells. There are generally 3 types of antigen markers, and they correspond with particular alleles, right? There is an I to represent what we call immunoglobulin. That is the name of that antigen marker. Now, if you have immunoglobulin A, you have antigen A, which is this circular looking antigen on your red blood cells. So that means all your red blood cells have this little knob with the circular top on it, and that tells your body that you have A blood type, right? It's how your body can tell that the blood belongs to it, right? And so your immune system reads it and goes, oh, that's ours, we're all good. If you have immunoglobulin B, it's an antigen marker of a different shape, right? And it's a diamond shape, for example, right? And that tells you that you have B blood type, right? If you have a lowercase I, you have no antigen markers, and so your body has none. Now, because there are 3 allele combinations there, that means that there are a total of 6 different combinations that you can get. The order does not matter. So you're going to see here, the order doesn't matter. It's just the combination, right? You can have 2 capital IAs, and that's going to give you blood type A, right? You can also have capital IA and a lowercase I, and that's going to give you blood type A still. Why? Because, if you get circles plus nothing, you still get circles. And so you're going to get blood type A. If you have IB, you get blood type B. If you have IB with a lowercase I, you still have IB because, diamonds and nothing give you diamonds. If you have IA, IB, you have both. You have squares and diamonds as your antigens, right? However, if you have 2 lowercase I, you have blood type O, which means you have absolutely nothing, right? Because nothing and nothing gives you nothing, right? So of the combinations, the order doesn't matter, right? So lowercase I, capital IA is exactly the same as capital IA, lowercase I. These two are exactly the same, right? So you have 1, 2, 3, 4, 5, 6 genotypes, but you have 1, 2, 3, 4 phenotypes. There are only 4 traits available: A blood, B blood, AB blood, and O blood. But there are 6 genotype combinations as a result of that. So this is a codominant relationship that also has complete dominance in its subtraits, right? So that's a complete dominance trait here, right? Because you have A blood type just because you have the dominant allele up to the top here. So you have a bit of a codominant and a complete dominance blend between the two, right? Now, why is this important? Because your immune system needs to be able to find out which cells belong to you. And so if you have A blood type, right, your immune system is reliant on reading these circular antigens to say that this belongs to you and it is yours. If you are B, your immune system needs to read B. If you are O, your immune system needs to read nothing, right? If you are AB, it needs to read AB blood, right? And it turns out the combination actually is different for different individuals, right? If you have O blood, Because your blood has no antigen markers, turns out you can give it to anybody. You can give it to another person with O blood, they'll take it very happily. You can give it to an A blood, and they'll take it very happily as well, because they're reliant on reading the A markers. So they're not going to bother whether or not you have no antigens on you at all. If you have B blood, that's also readable. And if you have AB, you can also take O blood. So O blood is actually the most versatile donation blood, because you, as an O blood, can donate to anybody. If you have A, you can donate only to other A's, or you can donate to ABs, because that immune system that's still going to read the A because it's got the capacity to do so, right? And so A blood can be donated to AB. If you have B, you give to B, and AB. Oh, sorry, I forgot to play this for you, right? And then if you have AB blood, you can only give to AB blood because of the presence of the A antigen and the B antigen. Your immune system needs to be able to read both, and so O blood, people with O blood, they're called universal donors. Whereas if you have AB blood, you are what we call universal receivers. You can receive blood from any individual, right? So I like to think of it this way, right? If you have O blood, you are very generous, right? You can give to anybody. If you are AB, you are you're very much a hog. You take everybody's blood, but you don't give to anybody but yourselves, right? So you're very selfish, right? Now, that's not characteristic of your personality. That's simply characteristic of your blood type. Now, there's a second attribute of blood, which is what we call rhesus positive and rhesus negative, which I'm not going to go into in detail, but in short, it's a much more simpler method, which is why, all rhesus negative blood can give to negative and positive. However, rhesus positive blood can only give to rhesus positive recipients as well. So that gives you a bit of an idea of how different blood types work. Okay, let's go to the next one. Now, when we talk about phenotype, that is the trait that is being exhibited. Almost all traits in an organism is its phenotype, right? Because whether it's the fruit color, the shape, or the seed count, or whether the leg length in your sheep, for example, they are traits. And so traits are the phenotype. Phenotypes aren't just visible characteristics, but can also include developmental and behavioral ones as well. For example, this is serotonin, right? Serotonin is a feel-good neurotransmitter. Your brain secretes it whenever you do exercise or whatever, and you feel good about yourself, right? Turns out, right, that's a protein, and that's made by the body, and different levels of serotonin in the body is a genetic trait. For example, mice with genetically low levels of serotonin are known to exhibit high levels of aggression. So, mice get really aggressive when you don't give them serotonin. And because serotonin is produced by the body, some people are going to produce more serotonin than others, and as a result become more, placid than other people, right? And so that is a developmental and that is a behavioral trait that is a phenotype or has a genetic cause as well. Now, not only that, your environment plays a really important role on the genotype. For example, right? Before we go on to the environmental influence, we can also talk about developmental. So this is penicillin. This is the effect of penicillin on bacteria. And you can see there that these are colonies of bacteria that are all reacting differently to the growth of penicillin. There's a penicillin, a tablet of penicillin, and then the bacteria is the white cloudy stuff, right? And then you can see that with every succeeding generation, different bacterial colonies are going to exhibit different levels of resistance against the penicillin. So the ones that are really resistant, like this one here, it's kind of pretty much grown over the penicillin. It doesn't really care. The ones that are not resistant, they are still maintaining a good amount of distance from the penicillin because it dies if it gets too close, right? That is a developmental trait of phenotype that has a genetic cause. All right, back to twins. So the environment plays an important role on, the exhibit of phenotype as well. How do we know that? Well, a few things. First of all, if you have the MC1R gene, you have the genetic predisposition for freckles, right? You are likely to produce freckles, and, you will probably produce freckles. However, the freckles don't appear unless they're exposed to UV radiation from the sun. So if you have the MC1R gene, but you don't walk out into the sun, you're not going to get freckles. And so that gives you an idea that, the environment plays a role. The second thing is in identical twins. Identical twins have exactly the same DNA. Why? Because they used to be the same zygote that split into two different eggs, and those eggs developed into clusters and individuals. However, it is known that identical twins will exhibit slightly different fingerprints. Why? Because the environment plays a role in the development of fingerprints, right? If the individual has, particular nutrient changes between one twin to the other, it's going to create different fingerprints. They're going to be very similar still, but you still get different fingerprints as a result of the environmental's influence, right? Which is, in this particular case, diet and development. Now, other environmental effects on phenotype can include, for example, PKU and diet playing an important role. PKU stands for phenylketonuria. It's a genetic disorder where the enzyme phenylalanine hydroxylase is not being produced by the individual, right? The PKU gene produces the enzyme PHE, phenylalanine hydroxylase, right? Phenylalanine hydroxylase breaks down the amino acid phenylalanine, which is PHE, right? Now, looking at the genotypes, if you have a capital P allele, you are producing normal PKU enzymes and your body works just fine. That's pretty much most of us. However, if you have a lowercase p, a recessive allele, that mutated PKU gene produces a non-functional enzyme. The enzyme doesn't work, you can't break down phenylalanine, and it builds up in the body, right? If you have a homozygous recessive genotype, lowercase p, that means that you can't do it and it's going to build up in the body, particularly the brain, and it causes a few different things. One, it produces this musty odor of sweat on the body. Two, it can create seizures because of the buildup of phenylalanine. You can create brain damage and result in long-term intellectual disability because of that buildup of phenylalanine. Now, however, just because you have the genotype lowercase p does not mean you have the condition phenylketonuria. If an individual with a genotype lowercase p can regulate their diet and is going to consume proteins that either have very little or no phenylalanine, you're not going to get the condition, right? Which means the condition can be controlled. The environmental influence, which is diet, can play a really important role in determining whether that trait is expressed or not, right? So if you have, lowercase p with a high PHE diet, you're going to get PKU. If you have the same genotype but with a low PHE diet, you have a normal phenotype. You're not going to exhibit any of those conditions, right? So early diagnosis in this particular case is really critical because the sooner you find the individual, the sooner you can then make alterations to the diet, control and minimize its effect. And there's lots and lots of different types of food that kids with that condition can have in order to regulate so they don't get PKU, right? Another example, so hydrangeas, right? Hydrangeas produce different colored flowers, but that's not because of their genotype. Turns out the genotype is exactly the same. It actually comes down to soil acidity, right? Soil acidity can change depending on the type of soil, and that change can still exhibit in different flower colors, right? So hydrangea cuttings from the same parent, which means exact same DNA, can have different flower colors depending on the soil acidity levels. If you grow it in 5.5 or less pH, you're going to get blue flowers. If you grow it in 5.5 to 6.5, you get purple, and then if you get 6.5 or more, you get pink. Now, why is that so? That is primarily due to the availability of aluminium in the soil, right? Aluminium makes it go more blue, basically, right? And what happens is anthocyanine, which is a protein that makes the flowers pink, right? The aluminium that is present in the soil is going to be absorbed into the plant, binds to the anthocyanine, and it makes it more blue, right? The binding of the aluminium to that protein makes it blue. Lower pH levels means that more of the aluminium is exposed to the plant. And so low pH soil means the plant can take up more aluminium from the soil, and then that aluminium binds to the protein makes it go blue. So you can see there, low pH, more blue, high pH, more red. Now, last section, I want to talk about epigenetics. Epigenetics is a very exciting new field where slowly scientists are learning more and more that not only is it about your genetic makeup, it's also about how your genes are being expressed or not expressed, right? So epigenetics is this kind of new area of looking at what happens if we don't change the genes themselves, but we change the way in which they're expressed, right? And so gene expression is, or the changes to gene expression is epigenetics without changing the DNA itself, right? Epigenetics is basically the interface between the genotype and the environment, much like a switchboard, right? So, for example, you go up to the wall and you want to control all of the lights in your house, right? Or all of the appliances in your house. Well, the light shining down on your house, that's the phenotype, that's the trait. the circuitry or the circuitry design and the switch design, that's the genotype. But if you walk up to the switch and you flick it off, right, then doesn't matter how well designed the circuitry is, you're not going to get the light popping up on the other end of the trait, right? In the same way, epigenetics is like the switchboard for all of your genes. Some genes are switched on, some genes are switched off, and as a result of the influence of the environment can actually switch your genes on and off, depending on how your body regulates it, right? Now, these molecular events can cause change, whether the gene, can change whether a gene is going to be expressed or not. And usually in epigenetics, the changes are temporary or reversible, but in some cases, it can also be intergenerational. So epigenetic, trait can actually be passed down to offspring, right? So, kind of the mechanism, how does it work? Now, this is a very complicated process, but in general, you have 3 different methods of epigenetics. You have what we call DNA methylation, histone modification, and then non-coding RNA, right? Now, DNA methylation is when you attach a methyl group to the cytosine, and that attachment of the methyl group there, the Ch6.3 over there, is going to switch off the cytosine, right? So cytosine being a nucleotide, and if you attach a methylated group into that, the cytosine becomes methylated, and it can't be read anymore. And if you can't read a C, it's like, you rub out, a quarter of all the letters in your sentence. There's no way that the particular gene can be read, and then therefore whatever the gene says is not going to be expressed, right? That's going to be very useful. Why? Because that by attaching a methyl group, if the cell itself can attach a methyl group to its genes, you haven't changed your DNA code, but by attaching this there, you're going to be able to switch off that particular gene in those particular locations, telling the body not to read that gene, right? And then when you remove the methyl group, it becomes readable again. So you can reverse that change as a result of that, right? So that's one method. The second method is what we call histone modification. You can add chemical groups that are going to cause DNA to wrap around the histones, making them unreadable, right? So, unfortunately, I thought that was an animation, but it's not, right? What happens is the histones, right? If you add a chemical group that's going to bind them all together, that DNA becomes no longer accessible. It's like if you, kind of tape shut your book. Doesn't matter how much, what you write in it, you can't open the book, and therefore you can't read the instructions that are in there, right? And that's another way in which the cell can then switch off certain genes or whole segments of genes as a result of that. You can also have non-coding RNA. So non-coding RNA can sometimes attach to coding RNA, and then it turns them off, right? So coding RNA for protein production can be attached using non-coding RNA, and that non-coding RNA is going to turn them off. So likewise, we talked about, non-coding DNA. Well, non-coding DNA can produce non-coding RNA that are going to switch off coding DNA and RNA, right? So that in the previous chapter, we talked about that, how your DNA can be switched on and off just because you have the genes. So let's give an example of epigenetics so you can understand, how it relates, right? So bees have a very complicated system, and they're kind of considered a bit of a super organism, right? You have queens, you have workers, you have drones, all sorts of things, right? Well, queens and workers are genetically identical. Why? Because the queens produce eggs that immediately hatch and not immediately hatch, sorry, that when they hatch have, exactly the same DNA as the queen. But the morphology, the behavior, and the physiology of the workers are completely different from the queen, right? Turns out that difference actually comes down to its diet. The queen eats royal jelly, which sounds very posh and very deliciously awesome, right? But the royal jelly is encased inside a little section here, and the workers, they don't eat royal jelly. They just eat plain old boring nectar, right? The royal jelly, which is pretty amazing, right? The royal jelly contains an enzyme, and that enzyme is going to inhibit the attachment of the methyl group, allowing the queen properties to form, right? The methyl properties or the methyl group attaches to the cytosine, and that's going to switch off the genes, right? So by feeding the queen royal jelly, the royal jelly has a particular enzyme. That particular enzyme is going to stop the methyl group binding to the cytosine, allowing all of the queen-like genes to be fully expressed. In your workers, they don't eat the royal jelly, which means they don't get the enzyme, which means that the methyl group are going to attach to the cytosine, and switch off the queen-like properties in those worker bees. So the worker bees end up being worker bees, even though they have the genetic information to be a queen, right? You can actually reverse this. Experiments have shown that by feeding worker bees royal jelly, you can actually transform them into queens, and they become viable. They exhibit a longer abdomen, and they can start laying eggs and all those kind of properties as well. Pretty amazing stuff if you think about it, right? And so, a lot of traits that we have are done through our epigenetics, right? Same genes, different levels of expression. Now, last one is the X inactivation. This actually happens in all females, right? In females, one of your X chromosomes are actually inactive in the embryonic stage. Despite the fact that you have two, one of them gets switched off and you only express one of them, right? If both of the X chromosomes are active, females are going to produce twice as many X-linked gene products as males. So all of the traits on the X chromosomes, you don't want double the amount of that. That's not exactly a good thing. Because that could overload the, whatever protein that you're producing in the body, right? So what happens is the body needs to switch off one of those chromosomes. And so the X chromosome becomes inactive and it's silenced by histone modification, right? It's repackaged into a heterochromatin, right? That X chromosome gets refolded and just kind of, just condensed into like a little suitcase. And that means it's inactive. It doesn't become red. That X inactivation can also be transferred down the germline throughout the life of an individual. And that means that, the cells in mitosis are going to produce X inactive cells. However, when the individual then produces offspring, those in that process of meiosis, that heterochromatin becomes uncoiled and into a regular X chromosome for meiosis, so that the sexual reproduction can take place. You can produce sperm and egg that are viable. Otherwise, half of your sperm or half of your egg are going to be inactive. Half of your eggs, because there's no sperm in females. Right? So that's reprogramming. You're removing epigenetic tags and reversing the X inactivation during sexual reproduction. So your body is kind of putting a tag to say, do not read, do not read, do not read. And then during meiosis, it reverses and reprograms that so that you can then read it again, right? X inactivation can have usually one of two results, right? The same X chromosome becomes inactive in all of the cells in the individual, or what happens is one X chromosome is inactive in half of the cells, whereas the other X chromosome is inactive in the other half of the cells. So, you can have two outcomes. So here's the examples A and B, right? The first example, right? That means that if you get rid of this X and it becomes inactive into a heterochromatin, right? This one's being expressed, and therefore all of the individual have this particular color coat, for example, right? Of that chromosome, right? On the other hand, if you have this one being turned into a heterochromatin, and you get blue as a result, right? So this could be one form of X inactivation. On the other hand, you could also have this as the X inactivation as well, where half of the cells in the body in early development becomes pink, and then or purple, and the other half becomes blue, and the individual has a kind of a mottled color of both as a result of that, right? If you look at cat coats, for example, right, the patchiness in certain calico cats are actually an exhibit of X inactivation. This means that in some patches the black is expressed, in other patches the orange is expressed, and then as a result of that, you get a blend of a mottled color or a calico colored in cats. Now, that is also why most calico cats are also female as well. You can have some males that have it, but mostly females. Now, this is a slightly different exhibition from codominant trait, and sometimes coat color is very complicated. It's not as easy as just all animals exhibit the same dominance pattern, right? But this particular case is a case of epigenetics, of X inactivation, whereas, for example, in cows, that is because of codominance, right? Okay, that's it. Hope you guys enjoyed that, and I'll see you guys in the next chapter.